decide how to change our stimulation, how we stimulate when and where, and then use the stimulation to modulate the brain state again. So this would be a closed loop.
Secondary measure of something that we think relates to brain state that we can detect with a sensor. And then we use a model of the brain to try to figure out what that biomarker actually tells us about current brain state.
We have an idea of what will be our target brain state, what we want to optimize for. And this can be, I want to see an increase in connectivity between particular areas of the brain. I want to see an increase in activity.
Think about optimization and how we might be able to optimize the parameters that we have inside the scanner. And this is what we call closed loop tES-fMRI. And that is what we are hopeful to see, how we might be able to have to combine closed loop tES-fMRI and combine all these things together to have closed loop tES-fMRI for the next step.
Where both a measure of coherence, which is basically synchrony within a band, or a measure of cross-frequency coupling, i.e. coupling from a low-frequency to high-frequency oscillation, have both been demonstrated to be responsive to clinically effective deep brain stimulation.
We would see how suitable this combination of brain stimulation parameters was for our optimization aim. So let's say if you want to increase connectivity in the frontoparietal network.
One thing to keep in mind is you want this loop to be faster than the change in the biomarker of brain state. Meaning if it takes us 10 minutes to go around, and at this point the brain state has changed, we've missed the target.
Relatively straightforward steps of individualizing the waveform, not yet in a time-dependent way, but even just participant, may truly increase the efficacy of these therapeutic paradigms we're developing. So this is recent work by our postdoc, Justin Riddle.
Hello, everyone. Good morning, good afternoon, good evening, depending on where you're based. We're delighted to welcome you today to the 5th webinar of the International tES-fMRI Network INTF around individually optimized non-invasive brain stimulation. So before starting the webinar officially, I just wanted to mention some housekeeping notes in order to have a more organized webinar. So the first thing that I need to point out is the video recording. So this meeting will be entirely video recorded, and we're going to share it publicly on the INTF YouTube channel as well as other social media platforms. So if for any reason you wish not to be in our recorded video, you may want to keep your webcam off during the webinar. The next thing is about your microphone and video. So please make sure that you keep your microphones muted during the entire meeting, because there are going to be some noises and people are going to be distracted. So whenever you want to ask a question or share any comments, you can turn your microphone on, and also you can keep your camera on during the entire session. The next thing is about the Q&A. At the end of the webinar, we're going to have a question and answer session, and we kindly advise you to raise your hand if you want to ask any questions or share any comments. And also, we highly encourage everyone to ask their questions and discuss their ideas around these topics. And the next thing, part of the GoToMeeting platform, please choose this chat box to ask your questions, post your comments or other related links to the topics, and make sure that you're choosing everyone as the recipient and not just the organizers. And the last thing is about any potential technical issues that you may face during the meeting with the GoToMeeting platform, with your webcam and other things. You can send a direct message to any of the organizers, and we will try to figure the problem out and help you with that. Enjoy the webinar.
Thank you, Panya. Ines, do you want to start?
Will do, yeah. It's great to have so many people here today. I can see, yeah, more than 100 people have just joined in, and it has been amazing to see the interest on this in Twitter, social media. So we were really delightful to have this webinar today. And I'll introduce, I was going to introduce Marom, but I think, Ahmed, do you want to say some words as well?
I'm really happy that we have right now we have 150, 116, so people are joining us, and that is great to see that level of interest in the community.
Great. So without any further ado, our first speaker today is Professor Marom Bikson, and many of you will know him and his work in the field of electrical simulation. He's a professor at City University, New York, and his talk today is entitled Closed Loop Simulation, Why Bother?
Thanks. Hamid, you might need to re-enable my ability to share screen. Is that okay? Great. Okay. Hello, everyone. I hope you can hear me. So I'm going to be giving a general introduction to closed loop stimulation because, again, we have a lot of really domain experts coming up, and we're going to be hearing a lot of detail. But I thought it would be useful to maybe try to frame the discussion a little bit. Hopefully, you can see the whole slide. These are my disclosures. Everything I'm going to be talking about today is going to be very generic. All right, so we need to start with the sort of, I think that there's a consensus that neuromodulation, when it's done best, is individualized, meaning that different people receiving neuromodulation for different purposes don't get the same dose, because if you give everyone a more customized dose, as individuals, as a group, people will do better. And that's obvious. TMS is individualized to motor threshold. DBS is customized to patient system response. And there's also a sense that if we didn't do that, things would not work as well. Now, in addition to customizing to the patient, we may also be changing it over time. So to the extent that we know that symptoms in many cases will evolve over time, and that supposedly involves a change in brain state, there's a notion that this dose may also be changing over time. I also want to clarify, that what we mean by dose can often be a little bit squirrely. I think it's most useful to define dose as what we give and not as how we got there. And so a classic example would be defining dose in terms of TMS motor threshold. That is a decision rather than necessarily defining what was actually given. Now, when we talk about titrating dose, you can see here, we can do it on different scales. Obviously, you can do it on the scale of an individual treatment. An individual comes in and they get prescribed their required dose. It can be done on a slower scale when we talk about clinical trials where we're trying multiple doses. That essentially is a dose exploration effort. And even on a slower scale, when devices are being designed to deliver specific doses, that is a dose exploration process that goes on an even slower time scale. And on the fastest time scale, we can have responsive stimulation. So this would be situations where the stimulation, the dose is being changed in real time based on some measurement. And I'm going to come back to this notion of time as we go through it. Now, when we talk about how dose is titrated, we'll talk about open loop, we'll talk about closed loop. So in a basic situation where, let's say, a researcher or a physician is evaluating symptoms and adjusting dose accordingly, that would be a classic sort of open loop approach. It is also very common to empower, or in certain technologies, it's common to empower patients to adjust dose. So this is true for most wearable technologies that are home-based, where patients are allowed the ability to adjust. There's an intensity knob adjust that they can use. Or we can have closed loop stimulation. So this is where there's a machine or there's an algorithm, and that is deciding how to titrate dose. And I want to emphasize when I'm talking about, we're getting into definitions here, but when I'm talking about open loop and closed loop, I'm talking about the decisions themselves. So for example, if you have a situation where a patient is self-reporting symptoms, but then that information is then being given to a algorithm to adjust the stimulation, that might be a closed loop approach. But if there is a human being but if we're using, let's say, an algorithm, but ultimately that algorithm is making a recommendation to an individual who's making a decision, well, now once again, we've inserted in a person who has now broken the loop. That would be my definition of it. Now, what do we base decisions on? The most basic thing to base decisions on is on the clinical endpoint, the thing ultimately that the person is receiving stimulation for. So for example, they're depressed and we're trying to enhance their mood, and so we're adjusting stimulation accordingly. But in addition to relying on clinical endpoints, there's often a reliance on these things that we call biomarkers. And it is useful to think about what we mean by biomarkers. As much as we sort of throw that word around, what do we mean by biomarkers? And I find it's useful to distinguish between two kinds of biomarkers. One is a biomarker of treatment response. So for example, you're trying to treat tremor, and a biomarker of that, a very direct one, would be an accelerometer measuring the handshake, right? So it is a surrogate, and it supposedly directly tracks the clinical endpoint. Or we're trying to treat epilepsy, and we're using a biomarker of epileptiform activity in the EEG. And so as one gets better, the other one gets better. However, there's another class of biomarkers that relate more to target engagement. So a great example of that would be adjusting transcranial magnetic stimulation intensity, let's say for treatment of depression, based on a motor response. Now, no one's going to suggest that making the finger twitch is what's improving the person's mood, right? We're using the finger twitch in order to adjust the stimulation dose as a biomarker that we are engaging the target. So that's a clear example where that's not a biomarker of treatment response, but of target engagement. In some cases, the line between these can get a bit blurry. If you look at spinal cord stimulation, where often paresthesia, tingling, is used to guide stimulation, if you believe that paresthesia is what's treating the pain, meaning it's sort of masking the pain, gait control, then that is a biomarker of treatment. It could be a biomarker of treatment response. But if you're simply using that to figure out where you are, which segment you are in the spinal cord, and now becomes a biomarker of target engagement. So I wanted to flag it up right now where sometimes it's not always clear which one we're talking about. And I also wanted to suggest here that the only reason why we would use any form of closed loop stimulation, any kind of algorithm, or even bother to use a biomarker, is to accelerate the dose titration process to reduce the cost. And that cost can be time, money, side effects. But the reason we're grasping for biomarkers is because there is in closed loop systems, is because there's a huge dose base, there's a cost in exploring it, and we want to get there faster. So that puts a sort of a wrapper over a lot of things. I also want to suggest that how we titrate is inherently linked to what we think is the mechanism of action. And I'll talk more about that, but I don't think we can disentangle those two. Because in whatever strategy we're using to titrate dose, we're often making a lot of assumptions about the etiology of the disease, what biomarkers tell us about the disease, and also about how the stimulation itself works. And though I'm talking a lot about treatment, many of us are also interested in neuromodulation as a way to probe nervous system function, to understand the brain. And here again, I think closed loop systems play a major role. I like to use the example of, for example, Hodgkin-Huxley and their foundational discovery that we still completely lean on as far as how voltage-gated channels work. That discovery would have been impossible purely by observation. That discovery was made possible because the nervous system was probed, it was stimulated, and through clever stimulation and monitoring response, we got to places where you couldn't get just by monitoring. So not just EEG, not just fMRI. We need to start poking the system around. And I think that's something we're going to hear more about from certain experts. Okay, a basic block diagram of how we could think about dose titration based on biomarkers. And so we have let's start here, we have the brain, and the brain has a certain brain state. We can think simplistically, healthy state, unhealthy state. But that's not necessarily apparent to us, right? We don't have direct insight into that. But what we can do is we can use a biomarker, such as BOLD, fMRI, alpha spectrum in the EEG, whatever it is, which we will detect via sensor. So in instrumentation and engineering, we might call the biomarker the measurand, but it's not the current brain state, right? It's not that it's a secondary measure of something that we think relates to brain state that we can detect with a sensor. And then we use a model of the brain to try to figure out what that biomarker actually tells us about current brain state, right? So we're not measuring brain state directly, but we're measuring some measurand, and then we're using a model to convert that into a prediction, essentially, of what we think the current brain state is. We compare that to a desired brain state, right? So prone to pain, not prone to pain. And if those things don't match, right, we would say, well, this seems like a good time to stimulate. If they do match, we won't necessarily. If they don't match, it's a good time to stimulate. We send that command to the stimulator, and then the stimulator then stimulates the brain. This stimulation is also based on what the stimulator is. Is it a deep brain stimulator? Is it a magnetic stimulator? And a mechanism of action, which is a model, right? We don't, this is a prediction, and then obviously we will then influence the brain. We will change the brain state, and then we can measure that indirectly via a biomarker. Again, we don't have access to true ground truth brain state, and then this loop is repeated. Now we can extend that to behavior as well. So I mentioned, for example, the example of tremor. So again, the brain is in a pathological, a Parkinsonian state that is reflected in a tremor that we can detect with a wearable wristband. That is a biomarker of the underlying brain state, which we can detect with a sensor. We use that to generate a model of the brain, which we then compare to desired brain state, and we repeat the loop. So I think this block diagram captures a lot of what is done with people, and it shows you where biomarkers sit. Now, just talking about electrodes, when the biomarker is electrophysiological, like a EEG or intracortical recording, we use electrodes as our sensor, and when the stimulation is electrical, we once again use electrodes as our actuator. So it's very convenient often to use electrodes. And the term BCI, when it comes up, I think traditionally would often come up when there was an intent associated with the sensing, but increasingly that's not the case. Just a few final points as I wrap up. We can obviously talk about forecasting. That simply, though I don't think often the term forecasting is, I think, used often more with weather than with brain, but the idea is still the same. We have information that is in real time, and we're using that information to make a prediction about the brain state in the future. And now we will stimulate accordingly. And so we have a situation here where I wanted to illustrate this picture, to illustrate that we have two essential models. There's one model happening that's looking at the sensor data, that's looking at the biomarker, and trying to make sense of it in terms of brain state. And we have another model where people talk about decoders, people talk about using various forms of machine learning to do this. And then we have another model here, which is the mechanism of action model, right? People will talk about input-output. People will talk about current flow models. People will talk about biophysical models, neuron models. These are all where these models sit. And so when we're trying to make this better, and this is I'll conclude here, I think we can talk about software innovation. And that happens, can we make this model better? And can we make this model better? Also possibly, I guess there's how this loop is made as far as control theory. And we'll also see a lot of discussion about hardware innovation. What is hardware innovation? Making the sensors more reliable, more increasing channel count, increasing focality, and so on. And going back to the concept of speed, which I think should come up all the time, again, when we're going through this loop, one thing to keep in mind is you want this loop to be faster than the change in the biomarker or brain state, meaning if it takes us 10 minutes to go around, and at this point the brain state has changed, we've missed the target, right? At the same time, there's many situations where we're throttled by our own approach. I mean, a great example of that is tDCS. So tDCS is a very slow stimulation. It's 20 minutes. It's typically applied once per day, right? So that means theoretically you could only go through this loop once per day, as opposed to 1,000 times per second with an invasive system. So we actually want to, in order to be able to train these models, ideally we want to go around this loop as much as possible. So let me finish there. I hope some of this was useful in laying the groundwork for what we're going to be here next. And I think we should go on to the next speaker.
Great. Thank you very much, Marom, for great presentation. We have the chat space, and people, if they are interested to ask any question, please use the chat space and put on your questions there. Then we can, then Marom, you can have access to the questions that people are asking, and then we can go back and discuss some of those questions in the last part of the talk. The next speaker would be Professor Til Ole Bergman, and Til, we are kind of the floor is yours.
Thank you very much. Great. So thanks a lot for the invitation. I'm happy to talk about brain state-dependent brain stimulation. And while this will be mainly in the context of TMS EEG specifically, a lot of aspects also translate into other techniques. And I'm happy to discuss the pros and cons of different techniques for the different approaches. To start with, for the last 30 years or so, we've been treating the brain as a black box. And when stimulating, for example, TMS, we get quite a variability in the outcome measures, whatever it is. Now, if we treat the brain as the dynamic system it actually is, and take the brain state at the time of stimulation into account, we can see that we get very different responses to the stimulation depending on the brain state. Each of those might have even a more homogeneous response, and so less variability in the outcome measures. And if we now would be able to know the brain state in advance, or actually at the time of stimulation, we could decide when to stimulate. And we could build closed loop systems here where we record a certain brain state, or we infer it based on certain imaging modalities like EEG or fMRI. And then we have certain real-time computations, making decisions, extracting the relevant parameters, and decide how to change our stimulation, how we stimulate when and where, and then use the stimulation to modulate the brain state again. So this will be a closed loop. What we've done in the past mainly is this open loop brain state independent brain stimulation. That is, we have a PC that triggers the stimulator, and that's it. The next stage, and that's what we mainly do at the moment, is brain state dependent brain stimulation, but it's still open loop. So we infer a certain brain state, for example, based on the EEG signal, compute in real time, make decisions, trigger the stimulator, for example, to characterize a certain brain state or to repeatedly interact with a certain brain state. But as long as we're not changing it, we do not have a closed loop. So for truly closed loop brain state dependent brain stimulation, we need to modulate with brain stimulation the exact parameter that we're actually monitoring, our target parameter, and help to bring it into the desired state and maintain it there. So what we've mainly done, including my own work in the past, is this open loop brain state dependent brain stimulation, which is already pretty interesting. And I want to quickly fly over two studies to give you examples. The first one was to investigate fast excitability changes during the sleep-slow oscillations. So during deep sleep with highly synchronized oscillations, the up states, during depolarization phases, the down states of the slow oscillation, reflecting hyperpolarization of large neuron populations. And we stimulate the motor cortex of sleeping participants here, measuring MEPs, automatically triggered by the detection of up and down states over the left motor cortex. And we could show, for example, that there's an excitability difference actually in every single participant between the up and the down state. Not only in MEPs, but also in the TMS evoked EEG potentials. So larger responses for the depolarized and for the hyperpolarized state at the stimulation site, and then also stronger responses distributing over the entire brain. And this is not an all or nothing phenomenon, but the size of the slow oscillation amplitude at the time of stimulation actually predicted the MEP amplitude, but only from electrodes close to the stimulation site. And the same is true, even though not here on trial-by-trial basis, but with bind responses for the TMS evoked potential. Another example now from the waking brain is the sensory-motor mu alpha rhythm. There our question was whether there's an asymmetric pulsed facilitation or inhibition going on, or maybe even a symmetric change in excitability. So we measured with a local EEG montage online the mu alpha algorithm and were either targeting a desynchronized state, so the lower 20% of the individual mu alpha power of the participant, or a fully synchronized state, so full-blown mu alpha oscillations. And while for the desynchronized state, we can only trigger at random phase, for the synchronized state, we targeted explicitly peaks, troughs, falling flanks, and rising flanks. And we measured single pulse MEPs to measure corticospinal excitability and GABA A receptor dependent short interval intracortical inhibition. And we made sure that we hit the right states. So you can see that mu alpha power was either not present, or it was clearly present in the low and high power conditions respectively. Here also shown with the TFR, we could show that we extracted with our local montage actually activity from the left sensory-motor regions and not from the even stronger occipital alpha. And most importantly, the phase targeting worked nicely at random phase. The time of response averages of 0, while we hit the peaks, falling flanks, the troughs and rising flanks pretty robustly across subjects. So what we found is that in the more excitable troughs and rising flanks, we have an increase in the MEP amplitude relative to not only the peaks and falling flanks, but also relative to a desynchronized baseline. While during the less excitable peaks, there was no difference to this baseline. This indicates an asymmetric pulsed facilitation happening in the presence of mu alpha oscillations in the motor cortex, at least. We did not see any effect on the SICI, and looking into specifically targeted power bins, we also found that the power of the mu alpha oscillation predicts MEP amplitude, so cortical excitability, also with a weak positive correlation. So we can test specific neurophysiological hypotheses by measuring excitability, characterizing certain parameters at different brain states. And we can also use this, and this work done by my colleagues from Ulf Siemens group in Tübingen. We can use this to induce plasticity. So here they targeted either the less excitable positive half wave or the more excitable negative half wave, the trough, with triplets of 100 Hertz TMS pulses. And when repeatedly targeting more excitable trough, an LTP-like increase in excitability was observed afterwards, while this was not the case when playing the same number of bursts into the less excitable peak. And we thought that this might actually represent a more general mechanism of phase-dependent plasticity that is also reflected in phase amplitude coupling, for example, slow oscillation spindle coupling during sleep, or theta-gamma coupling as known from the hippocampus, and that by repeatedly targeting with a high frequency input, whether from an oscillation or from a TMS burst, the more excitable phase of an ongoing oscillation, we can induce plasticity. So there are a lot of exciting possibilities for just these brain state dependent protocols, even though according to my definition, it would not be closed loop yet. But this is where we want to go, right? So how do we close the loop? And what do we need for that? So first of all, we need to go from our brain state to a certain recording, let's say an EEG signal. We have to process it in real time and then configure the stimulator also in real time to be able to change the brain state, right? And we know that a lot of different imaging techniques can be combined, and they can be combined with TMS, with TES, and also with focused ultrasound now. And each of these possibilities comes with certain challenges, technical challenges with respect to safety and artifacts, and maybe we'll hear a bit about that today and talk about that. But in principle, and that's important, it's possible to record brain activity with multiple methods and stimulate at the same time. And my very talented PhD student Umer Hassan has designed and programmed a toolbox for brain electrophysiological recording and stimulation, the BEST toolbox, which is an open source MATLAB toolbox that interfaces with many different recording hardware and software, and on the other hand, with different stimulators, a lot of different tMS machines, also focused ultrasound machines, in principle, everything that can be triggered by a TTL pulse. And we're always happy to include more devices. And it will also integrate robotized neural navigation in the future. And it's a toolbox that allows to design experiments and run them, get online results and proof of correct targeting in time, for example. And it allows to standardize a lot of the typical procedures in the lab, especially for tMS, but also for other techniques. And all of those procedures can be done in a brain state dependent fashion. So targeting amplitude and phase of certain oscillations, for example, using arbitrary spatial filters as you like. And if you're interested in, invited you to visit our website, best-toolbox.org, and also the upcoming webinar where Umer and I will introduce this toolbox and explain a bit how it can be used in principle to produce closed-loop brain stimulation experiments. So this is meant to facilitate this part of the of this loop. But there are a few remaining challenges. The first one is how to extract the brain state of interest. And here it depends a lot what you're interested in, of course. So considering simple local oscillatory states in the beginning, like amplitude and phase of certain frequency band, you need a good signal to noise ratio. Otherwise, you're targeting broadband noise that has been filtered. And you might need to even pre-select your participants to have sufficient SNR, because otherwise you can't target your brain state of interest. This might reduce generalizability, but it's required for proper targeting. You need to use adaptive frequency filters, adaptive power thresholds, take into account maybe the 1/f background noise, and use individualized spatial filters to really be able to extract what you think is an interesting oscillatory brain state in this specific case. It's a bit more complicated for more complex global brain states or network states when you consider synchronization between regions, connectivity, complexity indices, and so forth, because there are a few conceptual and computational limitations. Most of the computational limitations we will be able to meet with more stronger computers, but the conceptual ones are difficult. Some connectivity measures require to take into account a longer time period, and then when you're done analyzing or acquiring sufficiently long data strips, then the brain state has already passed. And for fMRI, it might be one of the particular challenges that the bolt response is very sluggish, and when we have our peak in the bolt response, the neural event triggering it, it's already gone by 5 seconds. So depending on the temporal dynamics of your brain state of interest, you might choose EEG or fMRI, for example, because you have to be fast enough to be able to extract it. But I'm pretty sure that we'll get there. Challenge 2 is the real-time removal of stimulation artifacts and confounds. And this is tricky. So especially for EEG or MEG, but I'm sure also for fMRI, there are a number of artifacts. And I'm not going into detail here, just listing a few of them. The problem is that for a fast closed loop protocol that is EEG based, for example, and using tMS or TACS, TDCS, TACS I think is worse, we basically go blind as soon as we switch on the stimulation. With already difficulties in tricky removing all those artifacts offline, but doing this online is a really hard job. And when we are able to do that, we're still left with all the somatosensory co-stimulation confounds. So TMS makes a click and you feel it on the skin. TACS can produce phosphines or subliminal retinal stimulation, cutaneous stimulation, etc. So you also have to deal with those. And so we need real-time artifact removal and appropriate control conditions if we want to go really closed loop and be able to quickly get the brain states. And then finally, so even when you imagine we could extract the brain state without problem, we're fast enough to change our stimulation and everything, then would we be able to actually control the brain? So I think the biggest challenge eventually is then that we don't know how to control the brain with stimulation. So again, for simple oscillatory states, we'd need to identify the stimulation parameters that enforce synchronization. And it could still be that the stimulation then evoked superimposed responses. It might be able to entrain or actually phase resets in oscillation, depending on the stimulation intensity. So we have to find out what exactly we need to do to change an oscillatory state. And for the more global network states, we have to identify the accessible nodes with the connectivity profile able to cause a state transition of interest. So we have to find out where to stimulate, identify the time windows of susceptibility for state transitions, so when to stimulate, and identify the stimulation parameters that enforce either state transition or stabilization, and if a state transition, then in which direction, so how to stimulate. Maybe control theory will come to the rescue and help us out to control these complex brain states with the simple means we have. And I'm sure that we're going to get there together, but there's a lot of homework to be done to be able to fully close the loop. So I'm thanking my lab members, my institutes, funding agencies, and thanks a lot for your attention.
Great talk, Til. Thank you very much. Thank you very much. For the interest of time, let's move to the next speaker. The next speaker would be Dr. Ines Violante, senior lecturer at Surrey University. Ines, do you want to start?
I would like, but I can't share my screen. I think you need to enable it. Oh wait, I can. So pleasure and a bit daunting to start after Til told us about all the issues that we have to face. And indeed, yeah, I was nodding so many of them, but I'm sure we'll get there. So the idea for today's talk is to give you some overview of a framework that we've been working on to optimize brain simulation with closed loop and real-time fMRI. And my lab is interested, so I just wanted to tell you how this sort of came, how I came to be interested in closed loop simulation. So we're interested in using non-invasive brain simulation to modulate cognitive function, and to use this to understand mechanisms, to investigate using brain simulation as a causal tool, investigate brain behavior relationships, and also as a possible treatment for a variety of neurological and psychiatric disorders that present with cognitive deficits. Okay, so how do I start? How do I started like I think many of us in the field of brain simulation with this idea that we have a particular problem. I wanted to modulate. I wanted to work working memory, and it's the idea that you have a particular task, and you sort of have an idea of which areas of the brain might be involved in that, and you use brain simulation as a way of targeting these brain networks with the hope that you will have an impact on behavior functions. And that was the case in a study that we've done where we've used TACS in two different conditions, and we did see that we could improve behavioral function. And we wanted to understand a little bit more about the mechanisms by which this was happening. So we've done a study, an MRI study, where we do simultaneous brain stimulation and fMRI. And this allowed us to see that indeed we could, these two different settings of brain simulation, either using, so we were stimulating frontal and parietal areas of the brain, and either feeding the stimulation in phase between this frontal and parietal area or out of phase, we saw different modulations of brain activity. And we also see different modulations in connectivity. So this is really interesting because we see, like many others in the field, we're seeing that you can modulate brain activity. This is somehow related with behavioral changes, and the effects of the simulation that we're using here, they are not restricted to just the site where you're stimulating, but they seem to lay over the networks in the brain that are involved in this particular task. And they seem to have these modulations not only in the magnitude of the brain activity that we're getting here with bold fMRI, but also in the functional connectivity between distant areas. However, as we start to look more at this, and we've done some studies in patient populations, we start to understand what Marom was referring to, which is this idea that you want to improve something in a patient population, but you're faced with this inter-individual variability, right, that we know that is there. And we start to see more and more factors that relate, that explain part of that inter-individual variability and how the effects of brain stimulation act on brain activity and on behavior. And some of those are participants' anatomy, connections, white matter connections between distant areas of the brain. We and others have looked at spectroscopy, and we are seeing that the concentration of particular neurotransmitters relates to how participants respond to stimulation. And others have also shown that there are genetic factors. There'll be environmental factors, age factors that would also play a role. And we start to think, well, how can we take all of this into account? So it's really important to understand the mechanisms here. But when you want to apply this, how can we take all these parameters into account? So these are like a big challenge here, right? This inter-individual variability. And the study that I presented you before is this sort of one-fits-all approach. So we picked a particular frequency, everyone got the same, right? Everyone got stimulated in the same place, everyone got the same intensity, and you start thinking, well, there must be a better way of doing this. And Til just show you really nice examples that if you look at particular brain states, particular rhythms in the brain, there is, you can indeed see that you can target this in a better way. But even if we are able to answer all of some of this or majority of this, and we have good models of how the brain works, and we understand all of these parameters here, right? We also then face it with this idea that, well, there's lots of different parameters of the stimulation that one can apply. We can think about different frequencies, different phases, different intensities. So all of these things just tells us that actually we have a combinatorial explosion. There's thousands of possibilities here, and there is no way that we'll be able to address this with our typical empirical experiments. And this is why we started to work on this idea of neuroadaptive Bayesian optimization, on the idea of closed loops, and in particular, we have been looking at neuroadaptive paradigms that employ active learning. And the idea here is that the algorithm that you're using in your closed loop stimulation approaches is learning in real time. So the stimulation parameters are being adapted in real time to the individual. So this cartoon here, the general idea is that for us, you have a participant with a stimulator in the scanner. We're collecting real-time old data, and this gives us an idea of how a particular area or networks or the whole brain is responding to a set of parameters. we have an idea of what will be our target brain state, what we want to optimize for. And this can be, I want to see an increase in connectivity between particular areas of the brain. I want to see an increase in activity. And you can think about this for other experiments that do not involve bold signal. It could be behavior, right? Like Marom was talking about tremor. And you have this machine learning algorithm here that is analyzing this data in real time and also proposing where to sample next in this big parameter space. And I'm sure Romy will tell you a little bit more about Bayesian optimization. So I'll leave that to her. I just want to today talk a little bit about how do you get there. In a sense, what Til was telling you, all those little steps on how to get there and how we are trying to get to this closed loop approach. So we recognize that having or understanding or having a particular brain state is important. We think that the parameters of simulation will be quite, that you apply will be quite dependent of what your participant is doing. So in our case, we are very interested in working memory. So we will have a spatial working memory task that we are introducing. We have fMRI, and we are using this, although it's a sluggish signal, it's true, but it does have some advantages. We have a good spatial resolution. We can look at particular networks that are of interest. In our case, they have to do with frontal-parietal networks, and the artifacts are minimized compared to EEG, so compared to doing simultaneous TACS with EEG. And for real-time pre-processing, we're using OpenNFT, and this is, I mentioned a lot of this work spearheaded by Tibor, postdoc in the lab, and he has been developing tools for OpenNFT as well. And we are using Bayesian optimization as our approach to optimize simulation for the individual. And as I said, we're using TACS in this case. So what are we doing at the moment? At the moment, we are focusing on actually collecting and analyzing offline data. So we are not in that closed loop scenario there. And why do we need to do this? We need to do this because with any sort of machine learning approach, you need some training data. You need to make some decisions about what would be your surrogate function. You need to tune your hyperparameters. And as Til was saying, there's many considerations to have here. So this is what we're doing at the moment. So getting our working memory task, collecting fMRI data, and having participants being stimulated in the scanner while they do this particular task. So this is just like a simple representation of the task, very classic working memory task. Participants see a grid, they're masked, and then they see a particular target, and they have to say if it matched the original grid or not. And we're doing this in a sort of repetitive match to sample because it gives us a little bit more behavioral parameters than just a one match to sample. And this is the sort of like montage that we're using at the moment. We're targeting these frontal-parietal networks. We have the lectures frontal and parietal regions of the brain. And at the moment, what we have is that all our participants are receiving intensities of 1 milliamp, so we're not tweaking this. The only thing that we are playing with here is a 1D parameter space, so a simple parameter space to try to define these functions and hyperparameters. So we're looking at just 4 different frequencies, 5, 10, 20, and 60 hertz. And what we can see is that participants do, so when there's no simulation, we see the pattern that we expect, where we see this activity in frontal parietal networks in the brain. And one of the things that we're looking at, that we sort of recognize from our previous studies and other studies, that the effects of simulation are not going to be restricted to one part of the brain. And we are indeed hoping to shape sort of network effect. So taking advantage of this network effect of brain stimulation. So we are actually using a multivariate approach. So we are using the decoding toolbox at this very early stage just to identify whether we can decode, where we can classify regions in this task positive network that respond to the different stimulation frequencies. So we have our 4 classes, and we're using the searchlight approach here with support vector machines, so a multivariate pattern analysis approach with a leave one out cross-validation. And we're having quite promising initial results looking at functional connectivity. So looking at functional connectivity between one of the areas underneath the frontal electrodes and the rest of the brain, where we can see a number of areas in the brain where we can indeed decode this activity, this response to different frequencies. So this is what we are now taking as our training data set, as data sets that we are going to use to close the loop. So my idea here, so the plan, as I was saying, is to go from where we are at the moment, right? We're still only looking at a sort of classic, okay, let's see which areas of the brain respond to stimulation. We're making sure that we identify robust parameters that will allow us to optimize. So we want to improve functional connectivity within this frontal parietal area, but we need to make sure that that's something here that we can optimize for, and that the brain is actually going to respond to these particular frequencies, because we do have the issue of how expensive these experiments are, right? How difficult it is to get participants for the experiments, and we want to, at least at this initial stage, make sure that we are optimizing something that we have the confidence that is generating different patterns in the brain, and is generating these different patterns in areas that we're interested on, and on a particular task that we're interested on. So the next steps are going to be closing the loop. So watch this space. And I think my general conclusions have to do on how we're doing this, our approach into neuroadaptive Bayesian optimization as a way of addressing inter-individual variability, and the fact that it's going to be really, really hard, if not impossible, to have a model of the brain. They just stop sharing from me. Okay, but I was just saying that our idea here is that we hope that this will allow us to address these issues with inter-individual variability, take this to closed loop approaches where we think that we'll be able to identify the ideal parameters to give stimulation in loco, right? And Romy will talk more about this idea that we believe that this is an efficient strategy to explore many more possible conditions that we'll be able to do in a standard experiment, right? You can imagine that if one is going to try all the possible plausible biological frequencies that you think would be useful, your participants will have to live in the lab for months until you test all of that with the repetitions that you want. And this type of approaches has already been applied in a number of fields. It has been applied in brain simulation, and it is extended to not just fMRI, right, but other types of data like EEG. You could optimize for behavior, you can choose your application. And we believe that this is going to be particularly relevant in patient populations where that inter-individual variability is even larger. And I just want to really acknowledge Tibor, that has been leading, spearheading the Bayesian optimization approach in my lab, and a number of other collaborators that have given such great input in the research that we're doing. So thank you all very much, and I'm sorry for the issues.
Great. Thank you for that great talk. As a reminder, I know there are questions being put into we'll take the questions to the chat, and I want to encourage everyone, not just the speakers, to join the discussion. It's a little hard when the talks are so interesting to also, engage in the chat, but let's try to do it all So thank you for that great talk. Moving straight ahead, I just want to introduce Dr. Lorenz from Cambridge, who will be presenting next on searching through the very large, that expression keeps coming up, explosive brain stimulation parameter space. Please go ahead.
Cool. Thanks for the kind introduction, and also I think a nice alignment of talks, because I will be talking about like in a bit more detail about Bayesian optimization and like the whole logic behind it. And yes, so just also as a pre-flavor, like Ines and me worked together before in the lab together. So not that you're confused. So I mean, I think Ines already like pitched the problem quite nicely, so I won't like talk about this in much more detail, but just as you know, like so far in group level studies, we're defining ad hoc a frequency and phase to simulate in, and then test this on a cohort of subjects. However, the biologically plausible range of possible frequencies and also phase differences for, like in this case, TACS is like large. So how do you actually choose like a frequency and phase? Like first of all, for a given research question. So for example, if you're interested in working memory, like how do we choose the optimal frequency to stimulate in? And the same though also, especially if you're interested in patient populations, they may vary due to anatomy and pathology. So the idea is to not pre-select those parameters before you start an experiment and like assume that also all patients or all subjects will have the same set of optimal parameters that would work for them. The idea for us was to basically define this kind of broad experiment space of stimulation parameters and then search through this in real time iteratively. And the aim is basically to find an optimal set of stimulation parameters. And as I said, we're using this approach that I developed over the course of my PhD called neuroadaptive Bayesian optimization. And it's a technique I validated not using brain stimulation, but like just in a standard experimental setting. So we were searching across like different audiovisual stimuli and tried to basically identify the optimal set that would like evoke some high activity in the visual cortex, for example. So we saw this is a very neat technique, and we could like exactly basically adapt this and instead search through like the space of like stimulation parameters. And the idea is to have, so it's basically, I think it's important to emphasize that our optimization aim is a bit different to the EEG people's optimization aim, as we're not trying to optimize on time. So we're basically trying to just, we're assuming a bit of stability of a brain state, let's just say, and then we're trying to actually identify the set of parameters that would optimize this one particular brain state. And so in that way, it's like kind of a block-based design where we would stimulate in a block, and then we would basically analyze the brain data in response to the stimulation. And then based on basically a certain connectivity pattern or like a bold activation pattern, we would see how suitable this combination of brain stimulation parameters was for our optimization aim. So let's say if you want to like increase connectivity in the frontoparietal network, we would see that like, for example, a certain set was not optimal. So what the algorithm will do, it will like explore the space more and search in an efficient manner. And how it does this, I will talk about in a second. But just as a general aim, and if you're interested in the technique, like this is, there was like a validation study that we published before without brain stimulation. So brief algorithmic introduction, as it is the core of the talk. So Bayesian optimization is a two-stage procedure. The first stage is, you can just think of it as a spatial regression. And it's a nonlinear regression, so we're using a Gaussian process regression. You don't really need to understand what a Gaussian process is, but just think of it as kind of an extension of a multivariate distribution to a distribution over functions. So it will be defined by a mean function and a covariate function. But forget the mean function now, we often set this to 0. So in that case, the Gaussian process will be defined by the covariance function. And the covariance function, so that's like now the clue to the whole issue is a set of basically functions that describe how smooth we expect the space to be. So this means if we're having frequencies aligned on X here, like in kind of a logarithmic fashion or however, we're assuming that frequencies that are close to each other will elicit a similar brain response, but frequencies that are apart from each other will elicit a distinct response. So this is like the similarity assumption that is encoded in the covariance function of the Gaussian process. And before we even collect data, we will define this prior, and we often do this by basically collecting pilot data before and tuning these kernels based on that. Now, once we observe data, so now those are brain responses in response to like certain, let's say, frequencies of stimulation. We're fitting the Gaussian process regression, and now you're seeing that the space of possible functions obviously gets slimmer because we need to fit those points. And what you nicely see is that we're not just now having like a prediction of the highest activation, but also of the uncertainty of this. So this just means that the algorithm is pretty sure that the functions need to fit through here, but it's very uncertain here. And this will be leveraged now in the second stage of optimization, which is the guided search stage, or where basically the algorithm closes the loop to propose which combination of frequency and phase it will test in the next iteration. And this is being done using a so-called acquisition function. And what the acquisition function does is basically leveraging this uncertainty. Let me just talk you through. So we have the same kind of 1D space than before. Experiments, so let's say frequencies on X, brain response on Y. Now our subject's brain response across all these different frequencies is our unknown function, which we don't know. So it's like the dotted black curve here. And we want to model that function, right? We want to understand what it is. So now we're observing 2 points, like we stimulated basically and collected data, and now we're fitting our Gaussian process regression in blue. And now what the acquisition function it does, in this case, it's the expected improvement acquisition function. It identifies the point with the high predicted mean, but also with the highest uncertainty. So where is the highest expected improvement? And this is here in this case. So we're maximizing the acquisition function, and this is the point where we sample next. So in the next iteration, we're repeating this. We're proposing, we're showing, or like we're exposing the subject to this frequency and phase stimulation. We're observing how the brain responds. We're refitting the Gaussian process. We're computing the acquisition function, again, maximizing, and that's where we jump next. And in this very like little toy example, you see that already after 3 clever iterations, we completely like fiddled, basically modeled the unknown objective functions with our surrogate model of the Gaussian process regression here. So that's the whole idea of this. And this is basically where the efficiency of the approach comes in. The efficiency comes from the fact that we don't need to see all frequency and phase combinations, but that the algorithm identifies the most informative ones, samples from there, and then can quickly move to spaces in the experiment space that seem more optimal, and then sample from there. So now we wanted to basically bring this into the closed loop world. However, you already learned there's so many different problems already. So we thought, okay, to show that the method itself is working, let's use like a scenario where we know ground truth. And this is like very difficult, right? So what we did is we used like actually a side effect or like a confound of stimulation. And this is like phosphene perception. So we said, okay, we know subjects see phosphenes. So it's an easy ground truth, basically optimization. But our algorithm doesn't know phosphenes, and our algorithm doesn't know which frequencies are basically inducing phosphenes. So we asked, okay, let's just have the algorithm identify the optimal tES parameters that maximize phosphenes. And just to briefly say, phosphenes are like illusory flash-like percepts that you see only at certain frequencies. So it's a very well-studied phenomenon, repeated like across many different studies. So it's a perfect ground truth functions for introducing a new algorithm to the field. So what we did, we spent like this large 2D space and we ran 2 studies. The first study basically searched across this white space. And then we saw that the algorithm was like you will see like the sampling was a bit shifted. So that's why we also zoomed in a bit further. And like in the second study, basically, we zoomed a bit further into and searched through this space. We tested this whole thing across 3 different montages. And we here did not neuroadaptive Bayesian optimization, but adaptive Bayesian optimization, meaning we didn't optimize based on any neural measure, because this is a tricky bit, and that's what Ines is working on now, right? So what we optimized on are simple preference ratings. So we just basically exposed subjects to 2 blocks of TACS, so coming from different sets of frequency and phase. So just going back, like you would select, like, let's say a frequency phase stimulation here and a frequency phase stimulation here or so. You stimulate subjects, and after this you ask them, where did you perceive phosphine stronger? So it was like a very simple binary rating, and we did this because we thought subjects are really bad and absolute, like rating absolute magnitude of anything. So that's why we went into this kind of preferential regime. And the nice thing is that there is a paper out there and like an implementation of Bayesian optimization based on binary observations, and that's basically what we used here. So it kind of transformed like basically the binary preference rating into a scalar, and then we can still model this Gaussian process. So if you are interested in the code, there is like this paper by Broch who basically introduced this Bayesian optimization, this particular Bayesian optimization algorithm. Cool. So now I already come to the results. I'm reporting group level results, but all the subject results are reported in the paper. And now you're seeing our results across the 3 different montages, the frontoparietal one, the occipital one, and then like a more classical one that basically only could optimize across frequencies because we didn't have 2 stimulations. And this was, all other papers always use this montage. So we used this again to just test it against the previous literature. And as you see, like just very nicely, first of all, as I said, the black dotted lines are basically where the algorithm sampled from. So you see the algorithm doesn't sample from anywhere, but from only the most informative points. And I think especially here you can see how it basically checks corners against like other combinations more in the center, and it will like quickly identify this whole space as not being optimal. So it will discard and focus on like sampling around the optimum. That's really where the cool efficiency of the approach comes in from, you know. And you see immediately that we have like strong frequency dependent effects of phosphine perception, and this has been reported in the literature. So oh yeah, just also this took like for subjects only 5 minutes for searching over 190 possibilities. And we do replicate these well-known frequency dependent effects. So a lot of papers reported 19 hertz, the lower beta range, as the range where phosphines are maximized for different montages. So you see that we're replicating this really nicely. However, what we're also showing for the first time is this phase-dependent effect of phosphines. So it seems that phosphines are also stronger when basically our simulation electrodes are in phase. And this is not super magical. No, instead, it's just like basically a feature of how we stimulate. And we could show this and like confirm this using computational modeling. So we had Ilkka Laakso from Finland on board who did like these amazing creepy plots for us. And he basically shows how the current density is for these different montages at different phases in the retina. So now it's well understood that like phosphines are a retinal phenomenon, meaning that like the retinal ganglion cells are stimulated and they basically fire for like certain frequencies more strongly. And you see that the current for like 0 degree is much higher than for 180 degree for both montages. And this really shows that it seems that phosphines are stronger for those phases. And this can be only declared due to like this kind of constructive interference for 0 degree phase shift, basically, when the waves superimpose. And next, what we did to also show basically, obviously, like this kind of objective function we had, like similarly looking to the sum squares objective function with like an optimum in the center is very easy to optimize for, right? So what we wanted to show is this approach also works for more complicated objective functions. So here we're modeling like 3 different ground truth objective functions. This again, very easy. Then this is like two optima here at the edges, and then we have like a very difficult like multiple optima objective function. And now you're seeing like different acquisition functions, and this one, like just like as a brief introduction, but this one is like one that favors more uncertainty, meaning it's more explorative. It will search more through the space, and this is a bit more exploitative. And in general, you see that this kind of explorative function works better, also obviously at the cost of like iterations, but it's very good in like mapping out the spaces. So you see that the Euclidean optimum, like this is the Euclidean distance of our identified optimum to the true optimum. And then this is the Spearman correlation of the whole prediction of the space against this. Oh, so, okay, I think I need to wrap up. Anyway, what I want to say with this is that the method identifies optima for more complex objective functions and also in the presence of noise. So we're also basically modeling this across different rate sensitivities. Just very briefly, so about the potentiality of the approach, we do think this technique has the potential to tailor simulation protocols to individuals. It can be used on behavioral or neural data. And also it could be extended to multidimensional spaces. So for example, to also optimize for simulation intensity or task conditions. So that's like, I think, an interesting avenue we also were talking about, to conjointly optimize task conditions and simulation parameters. And we just recently showed in a cohort of stroke patients, so this is this paper here, that we can optimize an individual patient and stroke patients, not simulation, but task conditions. So that's why I put it here. But it's a very promising approach that also could be applied to patient cohorts. That's what I want to convey here. And the Bayesian optimization is like a really fast developing field. And there are now basically new algorithmic developments to incorporate safety constraints into the optimization procedure. For example, for simulation intensity, you want to make sure that you're not burning your subjects in the scanner, for example, would be important. So considerations and challenges. That's like something Ines already pointed on. What neural measures should we even optimize for, and how should those be derived? Right now, we're doing this using like really elaborative offline studies. Do we have, I mean, for our method, it's really important. Do we have sequential effects, meaning that if you stimulate in one frequency and then the other has a different effect than vice versa? That's a problem. And also if we have carryover effects of stimulation. Again, in this case, we would try to optimize a moving target, which is like, in this situation, an impossible thing to do. Again, also as Til mentioned, do we have even sufficient CNR, contrast to noise ratio, to identify any or even the global optimum? If we're not identifying the global optimum, maybe this also has ethical considerations, given that we may be stimulating patients with like a suboptimal protocol. And what are even the controlled conditions to test any individualized protocol against? This should be probably like also talked more about, because it would be probably like a kind of group derived stimulation protocol, and then compare this against. Yeah, and then I just thank all these people involved in this particular study.
Thank you, Dr. Lorenz. Another really great talk. And again, some very precise questions in the chat. So please, if the speakers or anybody else wants to respond to these questions, please go ahead. Hopefully will be time for Q&A at the end, a live one, but in the interest of time, we move ahead and ask Dr. Widge. I think he's with us if he wanted to join us. And Dr. Widge has a very controversial title, suggesting that non-invasive brain stimulation has a lot to learn, and I believe it is true.
Dr. Widge did not pick that title. Dr. Widge will firmly blame Hamid for that one. And in fact, the title on your screen is the one I would actually describe, and really I think builds nicely on what Til was talking about, and is in many ways the invasive reflection of what in terms of what we're trying to do, of what he and others have been trying to do non-invasively. It appears that I don't see the slide transition on my GoToMeeting mirror for about another second after I click. So I'll try to slow it down a little. Anyway, As far as disclosures, I am going to talk a fair amount about invasive work. I am not going to specifically talk about any of the work that has been supported by Medtronic in terms of device donation, but it is there. Everything I'm talking about, the actual money for it comes from the US federal government or private foundations. The real disclosure, I think, is that I am going to tell you about stuff that none of this was done with my own two hands. This was done by an amazing team of people in my lab. It's a combination of both excellent engineers and some great neuroscientists, hardworking technicians, and some surgical and psychiatric collaborators who have given us unparalleled access to the inside of the human cranium. And so wouldn't be here doing this without them. As far as what I want you to walk away from, close your window with by the end of the day, is that, again, riffing on prior talks, that one of the more interesting targets for both invasive and non-invasive brain stimulation is not just the oscillations we've been talking about, but their synchrony, how they rise and fall together across regions, because this appears to be a fundamental mechanism of how the brain organizes cognitive networks. And at least with implants, it is now possible to measure that cortically, subcortically, even with current generation brain implants. And that, again, building on some of the prior talks, we've identified some specific methods for control of these synchrony features in a band-specific, circuit limit-specific way. All of this derives largely from a theory that Pascal Fries calls communication through coherence. Other people call it function through bio-oscillations. You'll hear the word ephaptic coupling. But the short idea being, and again, I think this riffs on previous talks, the idea that one of the things oscillations tell us about is the relative excitability up or down state of a given brain area, and that can organize cross-circuit communication according to Hebb's rule. Namely, if a neuron spikes at a given oscillatory phase and its downstream target happens to be next to an LFP that is at that same phase, it's more likely to trigger a postsynaptic spike. And by ordinary Hebbian plasticity, that is going to lead to these two regions wiring together, communicating better, passing packets of information along better. And there's a huge basic science literature on this idea, but the point I want to raise is that we are increasingly seeing this idea of connectivity and synchrony as important for circuit function in clinical diseases. This has been heavily demonstrated in Parkinson's disease, where both a measure of coherence, which is basically synchrony within a band, or a measure of cross-frequency coupling, i.e. coupling from a low frequency to high frequency oscillation, have both been demonstrated to be responsive to clinically effective deep brain stimulation. It appears that these same oscillations are present, at least in animal studies, of how psychiatric circuits work. So this one's from Josh Gordon, this one's from Katsuzarasa, this one's some of my own work from humans, which I'll get to in about 3 to 4 minutes. And so I'm going to show you today 2 quick examples. One is of how you can use that principle, that synchrony might be important, to provide one of those real-time brain state measurements we've been talking about in the chat, namely looking at the function of cognitive control or top-down response suppression, and then secondarily showing some of what we've been able to do with phase-informed neurostimulation to control these synchrony correlates in an animal model with a little bit of a teaser about human. So the first part of this, which is human invasive work, was done specifically in patients undergoing surgical monitoring for intractable epilepsy, which is a particularly interesting preparation because the standard way, if somebody has intractable seizures and you need to find a surgical focus to remove, the standard way to treat that is to place hundreds of individual electrodes throughout the brain with a heavy bias on instrumenting the mesial temporal lobe, i.e. hippocampus back there in green, amygdala there in orange, often a lot of prefrontal to premotor cortex, including things like subgenual cingulate and OFC that are really I'm not going to say impossible, but almost impossible to get after with EEG. And sometimes in some patients, you even get some basal ganglia, and those are particularly juicy. I'm not going to talk about it today, but I can put it in the chat. You go look, or you can see it on bioRxiv. We have a paper where we were able to use that subset of patients to demonstrate closed loop control of decision making. Here, oh come on, there we go. Here, what we focused on was asking the question, when is the patient attempting to engage this function of cognitive control, which again can be thought of as essentially doing what you wouldn't normally do, without delving too deep into it because it's a complex theoretical concept. And it is often tested with a variety of Stroop-like tasks where there is a response that would be highly prepotent, but you need not to make to answer it correct. And here, we ran multiple tasks in the same patients and tried to ask the question, can we look at correlations between multiple electrodes in each region, and can we look at how those correlated functions might be able to distinguish periods when someone's engaged in this effortful response suppression versus the periods in between when they're just resting, watching TV, doing nothing in particular, but also are not asleep and are not doing that sort of, the eyes closed or eyes open rest is a very different function than just kind of turning off your brain and watching daytime television. Why we're doing this is, as I mentioned, oh, seriously, come on, is because, as I mentioned, we have other results saying that if you know when someone is engaged in this cognitive control function, you can actually get them better through open loop or closed loop stimulation, which means it would be really useful to be able to detect, hey, when is someone in this state that they're trying to apply effort and they might need a boost? Think about this as taking them from having a bicycle to having an e-bike where you've actually got a force sensor on the pedal and are able to tell, oh, they're going up a hill, they're having problems. All right, let's turn on the motor just for a little bit. We're trying to build that same paradigm for psychiatric decision making. The thing I want to point out is, in fact, in this case, we can do that decoding. We can tell when this patient is trying to apply mental effort, but we can't do it with LFP power in any band from any region. That gray area is chance. The median performance, each colored dot is a different patient in this study. The median performance is completely consistent with chance. We can do it if we use a cross-regional synchrony metric. Then in this case, it's called canonical correlation or canonical coherence. We have other results saying that it's not sensitive to the specific metric. Well, that's not true. You can improve the performance if you optimize the metric, but you're above chance regardless of the metric. And what's interesting, by the way, is that this really crosses multiple bands. That there's a little bit of bias towards the low frequency theta band, which is classically associated with Stroop-like tasks, so that made sense to us. But that really, if you want maximal information, you've got to be looking at a multi-frequency classifier. That may become important as because the more bands you need, the more real-time processing you've got to do. So that may be an important system design principle. I can also comment that the performance was similar across two different tasks that we were using. These were the red and blue bars about 3 slides back. And that the network, so if you now look at this classifier and you extract the weights and say which edges are particularly important or highly leveraged in successful versus non-successful classification, you see comparable connections, not perfectly the same. One's a task involving faces, one's a task involving numbers, so we wouldn't expect it to be completely the same, but a couple things, including, for example, orbitofrontal to lateral PFC, and interestingly, frontotemporal and intratemporal connections, which are not classically associated with these tasks. This is something we're still trying to figure out, come up as highly weighted across both classifiers. And as another nice little control, only for faces do you start to see amygdala come into the picture very strongly, which makes sense given the role of that structure in face perception and also facial emotion perception. Now, if that's true, if I can tell you, hey, people say, in general, synchrony, coherence, cross-frequency coupling are important for cognition, and I can show you a specific example saying, yeah, that's true. They contain information that's not contained in the local oscillations, and that might be useful in order to do cognitive enhancement. This leads to the question that's been asked in the chat like 4 times. So can you actually target that? Can you come up with rational brain stimulation that can change synchrony, that can knock an edge in or out of these overall network operators? Wouldn't be talking about it if the answer weren't yes, we can. And now we're switching from humans to the other side of the lab, which is invasive work in rodents. And so what we've been working on is the idea that the way to do this is similar to how other speakers have shown, okay, let's modulate cortical excitability and modulate ERPs by phase-informed stimulation. Here we're doing cross-regional phase-informed stimulation. We're watching, in this case, it's the theta oscillation in one circuit and in one structure, infralimbic cortex of the rodent. And every time, not quite every time, we do leave a little bit of cycles in between for monitoring. But when we see, in this case, a trough of that theta oscillation, we're giving a single pulse, not a train, to a downstream region. The way we think about this is that what we do is we cause a phase reset downstream, and if we're repeatedly phase resetting downstream at a consistent phase of upstream, what that's doing is causing those two structures to learn to be in sync with each other. We're evoking some of the Hebbian mechanisms I referenced about 10 minutes ago. We targeted this particular circuit because there's some results from Josh Gordon's lab saying that this is a circuit that is particularly good at animals or particularly involved in a frequency that's particularly involved in rodents discriminating dangerous from non-dangerous stimuli. So we want to see, ultimately, can we use this to control anxiety, defensive-like behaviors that may have relevance for the use of this circuit in treating things like human post-traumatic stress disorder. Now, as other speakers have hinted, and I will further comment, this is easier said than done. Getting phase locking right is hard. There's the best toolbox. We have an open source toolbox that we call TORT that what we like about it is that it doesn't require MATLAB and you can plug it into a bunch of different rigs. It's also implemented in C++, so it's really fast. On the other hand, it does require to use a specific open neuroscience architecture. It's better suited to invasive than non-invasive, but it's another open source toolbox that is out there. I will comment that it took us about 3 years just to get this up and running. But when you do it, again, I wouldn't be talking about it if it didn't work. So 180-degree locked stimulation, there's the theta band highlighted, can specifically create interregional coherence. I think this thing over here is a harmonic. Can specifically create interregional coherence in the target band. And if you do the opposite, if you enforce a uniform distribution by only stimulating the amygdala at arbitrary phases, every stimulation comes on a different phase of the controlling frontal oscillation, you can do the opposite. You can take an edge out of the network. Also, consistent with what I was saying about neuroplasticity, you get outlasting effects. So the way we've been doing this is a 30-minute protocol. We stimulate about every 5th theta cycle, so about once per second, total of about 1,000 pulses. And we can see substantial effects that separate from baseline, in some cases out to 90 minutes post-stimulation with the animal just free behaving. We haven't seen any across days carryover effects if anyone's thinking of that question. But this is interesting because it certainly implies that this could be a paradigm useful for non-invasive stimulation where we think about the paradigm of bringing someone into the office every day for a month to try to staircase up neuroplasticity in a circuit over time. I will comment that we are in the process of demonstrating that this works in humans. This is a collaboration with Joel Voss at Northwestern, where one of the nice things about this closed loop pipeline is you change one piece of code and you can run the exact same thing that we run in animals on a human grade rig for those epilepsy patients. And in fact, owing to some differences in how the network communication protocol works, the human system's actually better than the animal system. Now, we've done one patient so far, and that patient didn't have amygdala electrodes, so we're looking at hippocampal cortical connectivity, but we did target, again, the theta band, and we were able to see statistically significant differences and a specific increase in that incoherence in that band. Now, a bunch of stuff happening down in sort of sub theta as well that I don't know whether that's going to be idiosyncratic to this patient, if it's going to continue to show up, if there are things I need to explain about how this circuit or how humans work differently. So super, super preliminary. But I think the fact that this also shows how you can use an invasive animal model to identify things that you then might want to replicate in a human, where you often have much less time compared to, you can keep a rat in a box and test it for a month if you need to find at least a narrowing of the stimulation space. This, again, builds on some of the optimization theory we've been talking about, in the sense that this suggests that we could at least use the animal work to create strong priors to constrain the human search space a bit so that it's not quite so infinite. Now with that, where are we going next? Our big focus now is on saying, all right, locking this stimulation to oscillations can alter synchrony. Is that useful? I highlighted that this is a circuit that's involved in safety danger discrimination. So we are starting to deploy these techniques in a rodent model of pathologic avoidance, where the animal has to choose between sitting on a platform where it's safe from a signal foot shock and going and getting food, which is the only time it's able to get food is when it risks having a shock delivered. And so we consider this a reasonable assay of risk reward discrimination, and we think we should be able to bias that by messing with frontoamygdalar safety signaling. The other thing we're trying to do is to say, how could you take this, and since we're primarily an invasive lab, how could you take this and make this into a implant-friendly signal processing algorithm, because it is certainly way too power hungry right now. So we are collaborating with a group of electrical engineers in Switzerland to actually push all of this into an application-specific integrated circuit that would enable running these same phase-aware algorithms on an implant. Currently, and I will tell you, I've tested a lot of what's out there, you cannot do this with any extant deep brain stimulator or anything else, and boy, would we really like to be able to. I will comment that we also have thought about, could you build such a smart chip into an EEG system so that you've got an EEG system that will automatically give you phase information on any channel or montage you want? Answer, maybe. We're in discussions with EGI about whether that's useful to them, and ask me again in about a year. And with that, I know we are doing questions mainly in the chat, and we're certainly sort of creeping time, so I will hang around, and I see there's stuff that's come in, and I will reply to it.
Thank you, Alik, for that great talk. Moving right ahead, Dr. Frohlich of the University of North Carolina will be speaking next. His work spans the smallest cells to the biggest diseases, so I'm looking forward to hearing his talk.
Thank you very much. Good morning, everyone. It's a distinct pleasure to be here today, and hopefully we're about to see my slides. So here are my conflicts. I just want to briefly mention that I've started a company that is heavily invested in closed loop control of brain activity. You can ask me questions about this later if you have any. Also, I want to start with the acknowledgement slide, because as other speakers have also emphasized, none of what I present today is my own personal work, but has been the work by an amazing team that I'm very privileged and honored to mentor. Also, this is a pre-COVID picture, so hopefully soon we get the ability to take a new picture. Anyways, just to clarify, because I will be talking about disease states, I'm a researcher, not a clinician. None of what I present today is medical advice. Most of what I'll be talking about has not been demonstrated to be effective to the extent that it would be FDA cleared. And as always, I'm sure we can agree more research is needed. When I was asked to give this talk, I was thinking back to when my interest in closed loop or feedback control started. And I had some memories come back from my time as an engineering intern at a company that was building these huge printing presses to print newspaper magazines. And as it turns out, there's a very sophisticated process with a lot of control engineering, a lot of measuring of different aspects of that process of the paper being fed through the machine, and little mechanical adjustments that are made in real time to ensure that the paper that goes through the machine doesn't break, because if it does, there's a significant downtime with according economic consequences. So that's what you learn about in electrical engineering. And in a most simplified form, and we've seen this in earlier talk, the way how to think about this is pretty straightforward. We have some kind of system that we want to control, and typically in engineering, we have a model that describes in a quantitative way the dynamic behavior of that system to input, and all we need to do is measure the output of the system and compare it to what we want it to be, and then feed according input back into the system. And the controller is essentially the algorithm that determines what that input should be in real time as a function of that discrepancy between desired and actual state. And quite a while ago, when I was still an undergraduate in electrical engineering, I thought, well, the same principles could also be applied to neuroscience. And here this plot shows an example. This is a simulation of a state-based controller that worked very well to suppress repetitive neural firing in a model of a biophysical model of a single neuron. And in very optimistic terms, I concluded that essentially control engineers called to action should consider the manifold of interesting and challenging problems arising in the design of sophisticated electrical stimulation devices for rehabilitation of patients suffering from dysfunction of the nervous system. And here we are about 16 years later, and I think what this event nicely puts together here is that we've made quite some progress, but that the actual vision that this is a treatment that works is still something that a lot of work is needed. So as I was shifting my thinking from individual neurons to networks, we and many others realized that networks can be entrained and modulated, and we heard a lot about this today, so I'm going to go fast here in the interest of time. But what really puzzled me is how little we've been thinking about the fact that the brain not only generates electric fields, but also, as I've just shown you, that neurons and networks of neurons respond to electric fields. So that's another closed loop system. And perhaps we can make the distinction to aphatic coupling, which is really more a cell-to-cell bleeding over of changes in the electric field. But here, I'd rather would like you to think about a meso to macroscopic phenomenon where we have this built-in feedback loop that whatever a population of neuron does, necessarily in synchrony to create a sufficient strong electric field, that very field feeds back onto neurons. And originally, my interest here was in terms of understanding this as a mechanism, as an active communication mechanism that enhances synchrony in the brain. But ultimately, what I would like to share here with you and state is that these principles that we're talking about today have essentially already been in some ways built into the brain, and by studying those, I believe we can derive interesting and novel approaches how to look at that. So here's an in vitro early example of this idea, where we benefited from the fact that we can record action potentials in a slice preparation and essentially create a mesoscale simulated electric field. And what we found when we essentially closed this loop, that an ongoing kind of rhythmic oscillatory network pattern becomes much more crystallized and regular in its rhythmic structure with this feedback mechanism built in. Now, one thing that we've heard over and over again, and I'll get to as well, is obviously having the benefit of being able to record action potentials that occur in a very different frequency bands from the stimulation waveform applied is a benefit that we don't have in human non-invasive applications. But in fact, what I had tried back then was to build a control system that modulates the endogenous field generated by the tissue preparation. And it was by far not perfect in terms of the ability to control, in this case, suppress the endogenous electric field, but it was partially successful, and already that kind of imperfect control demonstrated how suppressing that or turning off that feedback loop affected the oscillatory structure of the network that we targeted. So that sounds quite straightforward and should be easy to implement in reality, but as we've also heard from previous speakers, we do not have these models that we have for printing presses and cruise control and other very successful control engineering applications. So I've been carrying with me, and I think other engineers in the field as well, a certain sense of disappointment and perhaps even disillusion that the control engineering, the way how it's been developed to control man-made systems, only do not have a direct and complete successful translation to the control engineering problems we're talking about when we talk about closed loop brain stimulation. One of the reasons for that, of course, is the highly nonlinear nature of the system across scales, right, from individual ion channels to the electrical signaling in cells, all the way to entire networks. And then finally, what others have mentioned too, I think that's very important, and this is not a true equation. I hope I don't upset the physicists too much in this call, but essentially there's this trade-off, right? We can stimulate at whatever amplitude, whatever way we want, but our ability to in real time recover the signal is very low. Vice versa, of course, we can record with quite sophisticated strategies and decode according signatures, but that only really works in the absence of stimulation due to the stimulation artifacts. So how do we go on from here? And what I want to share with you is kind of a very pragmatic, straightforward way to think about this, which I hope will accelerate the translation towards new treatments, because that's where my vision and passion is. So the obvious approach that we've already heard about implicitly and explicitly, which I feel is really worth pursuing, is a simple interleaving of measuring and controlling or stimulating. And that solves the artifact problem. And assuming that the stimulation has more than just an instantaneous effect should work, though there are some time delay challenges that I believe the field needs to address. Many of us, and as we've heard today, in very smart and sophisticated ways, are thinking about how to create empirical input-output maps instead of mathematical control algorithms. Also, the state dependence, whatever control algorithm we develop, the state dependence essentially adds another layer of complexity that we need to take into account. And then finally, models of different kinds are all important in this field, but going back to the biophysics of cells and networks, I would like to suggest is really helpful to understand how the stimulation actually works on the way towards developing a closed loop approach. Now, just very briefly, some really simple, straightforward initial steps that are not yet that real-time closed loop control of brain activity we're all dreaming about, but might already bring along quite a benefit in terms of target engagement, is probing very simple relationships between stimulation parameters and the response of the network, such as the response to TACS simulated here in a large-scale cortical network model as a function of stimulation frequency and amplitude that helped us in the field to identify target engagement principles about the potential benefit of tuning the stimulation waveform to specific features, in this case, the oscillation frequency of the target oscillation. Now, this is an insight or a hypothesis derived from a still very simple computational model, but you can understand the importance of that intuitively by looking at this figure where a matching of stimulation frequency has an augmenting, synchronizing effect, versus a detuning or a difference between stimulation frequency and stimulation and network frequency could have actually the opposite effect. So monitoring that and adjusting the frequency is a first simple, straightforward step that already may bring quite some benefit. Now, does this actually occur in reality? This is a very recently published paper in Nature Communications where we look at whether we find that Arnold tongue or that inverted triangle of target engagement in an intact brain, which we have. So then the question is, do the preclinical animal models fit together with the computer simulations, which we confirm here through this more sophisticated thalamocortical model. So at that point, it's a very basic, straightforward principle to think about the stimulation frequency and adapt the stimulation frequency in a need-based way. So more recently, we've adopted this principle for our human studies, and this is a single session target engagement study in patients with major depressive disorder, following up on our previous RCT on TACS for depression. And we've inserted that additional step of identifying individual peak frequencies and adjusting stimulation accordingly, with the goal of creating a more consistent effect of stimulation, as demonstrated here in this topographic plot. We're able not only to replicate, but also actually increase the effect of even a single session of stimulation on the target, and the target that is the desired target is a decrease in left frontal alpha oscillation. So that's exciting work that demonstrates these relatively straightforward steps of individualizing the waveform, not yet in a time-dependent way, but even just participant, may truly increase the efficacy of these therapeutic paradigms we're developing. So this is recent work by our postdoc, Justin Riddle. And the reason why we're interested in that is because recovery from a psychiatric illness is a process. So what you see here is from a case study from our group where we deliver TACS weekly, and you see symptom changes based on clinical assessments to the left and right based on self-report. So you see all these nonlinearities and all these steps, and presumably, accordingly, we'll see dynamics, longitudinal dynamics of changes in network activation and dynamics, and tracking these feature adapting stimulation. Again, I'm talking about closing the loop in terms of rational algorithm-based adjustments, even perhaps on a time scale of week to week, may further accelerate the recovery process. Here's another example of a schizophrenia patient who received 20 weeks of weekly TACS. And again, just to illustrate that there are these slower changes in symptom manifestation. And what I would like to suggest is, while we're working on this idea of millisecond precision closed loop control, which is certainly most very exciting, there are also slower timescales of thinking about how to adjust treatment based on underlying change of network substrate associated with recovery. And we have some evidence that indeed symptom recovery is associated with change in oscillatory features, including the frequency, as from our recent trial of TACS for auto hallucination schizophrenia, where the stimulation shifted the frequency to the target frequency that then drifted back over time. So tapping into these mechanisms and creating dynamic ways to adjust the stimulation frequency presumably could further increase the efficacy of these treatments. And finally, just very briefly, zooming in on faster, kind of more real-time, but still in this way of alternating recording and stimulation approach, a study from our group where we detected sleep spindles, these transient thalamocortical oscillations, and applied waveforms matched to the sleep spindle in this dynamic closed-loop way. And essentially, what is I find interesting about this study is that because these sleep spindles occur transiently, this idea of having to measure and stimulate and wait to measure again was actually not a big detriment to the overall control goals. And indeed, we were able to demonstrate that stimulation versus sham increased memory consolidation, the way we suspected sleep spindles would augment that process. But we're also able to demonstrate essentially on the subsequent sleep spindle a frequency-specific enhancement through that stimulation. So, in other words, what I would like to leave you with, these principles from very sophisticated, complex, very theory-driven, on one hand side, we've heard about that today, all the way to the very pragmatic, incremental improvements of how we listen into the brain, adjust stimulation. I believe as that spectrum all comes together, we'll be able to develop the next generation of effective non-invasive stimulation treatment for psychiatric illnesses. whatever the underlying idea is, I just want to emphasize that the idea of thinking about the targets carefully, identifying them, besides thinking about the target engagement, the design of these closed-loop algorithms is equally important. And finally, what I would like to encourage the community to do is at the same time to rapidly translate these approaches to at least feasibility trials in humans, given the incredible safety profile of non-invasive brain stimulation, since we're facing an unprecedented mental health crisis. And I believe I and many others of you also believe that these improved targeting strategies could be of therapeutic benefit. And I'm looking forward to us as a community advancing this and helping patients as soon as we possibly can. With this, I'm at the end of my time. We've been continuously and generously supported by the NIH, and we have new postdoc positions to fill. So if that sounds like a profile you're interested in or you're matching, please don't hesitate to reach out. And I would like to thank you for your attention.
Okay, great presentation. And now we are almost at the end of the webinar. So I have prepared something like a 15-minute talk, but I'm gonna kind of make it really fast to be able to make the deadline of timing that we have. Also, we will still keep receiving questions on the chat space, and we try to kind of get to some of those questions at the end of the webinar today. Okay, we have been discussing about kind of how we might be able to get to a level of closed loop tES-fMRI, and I'm starting from this part, discussing that, okay, in my lab, like many other labs, we do the same sort of one-size-fits-all approaches with one frequency stimulation in frontoparietal connectivities, trying to see how we might be able to modulate kind of brain activations. But we know that it is not optimal with all the differences that we have among subjects. And then the question is, okay, is there any chance that we can get to an optimum level? What we are expecting to have, what we really like to have. So we like to have the kind of brain stimulation inside the scanner in a way that we are going to be able to record what is happening right after a stimulation or during a stimulation. So this is what we call concurrent tES-fMRI. And the good news is that process is moving forward. We have recently published a method paper that discusses about different potentials for integrating TDCS and TES with fMRI, and some technical notes if you are interested to do that. And then we have recently published a systematic review discussing about something like 130 different tES-fMRI studies, that some of them, something like 34 of them are concurrent tES-fMRI. So discussing about the parameter space and the outcomes that people have tried to do so far. So it seems that the field is moving forward. And then within the international network of tES-fMRI or INTF that you are already in this webinar, we tried to make a sort of a consensus paper about the methodological details that people are supposed to consider when they do concurrent tES-fMRI. And this checklist is right now under review in Nature Protocol. So we are hopeful to be able to have it out as well. But you can find the checklist in medRxiv. And we divided the technical challenges for that and kind of minor details that people need to consider into 3 main categories. And there is also a methodological checklist that people can use when they want to design a concurrent tES-fMRI to be able to get to the fine-tune kind of optimization of what they want to do. Meanwhile, after having concurrent tES-fMRI, what we really like to have is just to see how we might be able to adjust parameters that we have. Is there a chance that we can measure the level of target engagement, and then based on the level of target engagement that we have, think about optimization and how we might be able to optimize the parameters that we have inside the scanner. And this is what we call closed loop tES-fMRI. And that is what we are hopeful to see, how we might be able to have to combine closed loop tES-fMRI and combine all these things together to have closed loop tES-fMRI for the next step. For being able to do that, we are right now working on a setting with postdoc and Aki and a PhD student, Benny, and they are doing a really nice job in putting all these things together. And of course, like many others labs, we have started with phantom stimulation, kind of watermelon stimulation, using different kind of methods to make sure that we can have a reasonable level of signal, and then we move forward with human subjects trying to see how we might be able to actually do tES-fMRI in a reasonable way that we can have reasonable level of signal, and kind of try to see how we can optimize the way that we are doing this stimulation. But let me just kind of give you some ideas about some of the challenges that we have, and then I'll kind of open the floor for those who are interested to discuss about some of the challenges that we have for this specific tES-fMRI closed loop stimulation. One issue is about electrode placement. It's just not as simple as thinking that, okay, we are stimulating for the parietal, we put an electrode on the frontal side and the parietal side. Things are way more complex in terms of when we want to make sure that we are doing an optimized electrode placement. We have done a really nice with the PhD student Ghazal Soleimani, and she did a really nice job in terms of finding a specific computational pipeline to optimize the way that we are putting electrodes for network level stimulation. And people, if you are interested to that, we have recently put that in bioRxiv, so you can see the way that we developed a specific computational pipeline for optimizing the way that we are putting electrodes for frontoparietal connectivity measurements and stimulation. And we showed that these optimized way would be able to get into the network that we are interested to, but those non-optimized ways might not get to the level of connectivities that we are interested to have. And even think about the kind of the network level stimulation and how we get to different networks, because even when we talk about something like prefrontal stimulation, we know that in prefrontal stimulation, we can get to executive control network, we can get to saliency network, we can even get to default mode network with just simulating the prefrontal cortex. So we need to be careful about how we optimize the electrode placement. And other than just computational models, there are some technical issues in terms of using electrodes inside a scanner in a way that would be feasible. They should be small. They should not be on the back of the head because we put the kind of the subject inside the MR code. So there are some technical challenges that people need to think about. And we have tried to solve some of those technical challenges, trying to see how we can kind of put the electrodes inside the scanner in a way that would be functionally feasible. And then you start to get into the technical challenges in terms of how to design a study. As other speakers have mentioned, there are many ways of kind of doing these optimized stimulation or closed loop stimulation, and try to define the arms that you have inside the study, the ways that you kind of do the sequence of different trials inside the study, and even trying to optimize the way that we are putting these kind of sliding windows of kind of recording and stimulation, and how these kind of processes are working together. And even in terms of thinking about the ways that we can make control interventions, there are many different ways that you can define control intervention. For example, here I'm showing you 8 different ways of having a control intervention for the active stimulation that you are developing. Those who are interested to those topics, we have recently put together a protocol paper with all those technical details, which is right now available on bioRxiv. So you can see all the technical details that we have been struggling with, and the decisions that we made, and the kind of the reasons that we had behind those decisions. And then you get to the level of, okay, what is the main outcome measure that I'm interested to put in my kind of pre-registration protocol, and see what are the things that I'm looking for in terms of the directionality of changes or the absolute values of changes, the behavioral outcome. There are lots of challenges on that side as well. And then the third challenge, as other speakers have mentioned, temporal dynamics of brain response is something that is really complex, and we know that is the case, especially for the fMRI analysis. People have not been exploring that very much. We have recently published a paper in Neuroimage discussing about the temporal dynamics of response, and we know that there are areas that they have a sort of a stable response over a specific fMRI task, but certain areas they do not have kind of a stable response. They have a dynamic response over the time. For example, in this study, we explored a discovery sample, and then we had 2 replication samples, and we showed that this is something that we can replicate in terms of the temporal dynamics. And we showed that if we replicate that, we do the testing again in something like 2 weeks afterwards, the response would be different in terms of the dynamic response. So there are challenges for temporal response. And then considering those temporal challenges, we need to start to think about, okay, how we can define the sliding windows, how we kind of define the pairs of stimulation, and how we might be able to tackle in those challenges, and then getting into the optimization kind of protocols. Those are going to be complexities that we are facing. Okay, these are my last slides. We are trying to put together something like a white paper discussing about the challenges and major kind of questions that we have and major decisions that we need to make when we design a closed loop tES-fMRI study. We are already have ongoing trials with this kind of this setting. We are reasonably happy with the level of signal that we have, and I will be happy to kind of share the experiences that we have with those trials with you if you are interested. Those who are interested to kind of continue the interaction with us in this network, we have a YouTube channel that we put all these webinars that we have on that YouTube channel. And Payman, who is right now here with us, is managing the kind of the YouTube channel and lots of other things that we are doing today. So many thanks to Payman for doing this in a really neat way. We have a network of people around the world, and the ideal scenario for us is just thinking about how we can bring people together and try to see how we can help each other in different labs around the world to be able to make this happen. Because it takes a village to make this happen. And we are hopeful to put people together, share protocols with each other, and try to see how we might be able to have multiple labs involved in these concurrent tES-fMRI studies, and then moving forward for something like a closed loop tES-fMRI consortium collaborating on that dimension. So that's the ultimate plan that we have. And just the last slide, the next webinar we are planning for, we are going to extend this idea of some sort of optimized stimulation with computational head modeling, combining computational head modeling and fMRI on September 23rd. For those who are interested to join us, please keep in touch. You can send us an email, and you can follow us on Twitter. So, Marom, I try to be really kind of fast, so hopefully we can let me just kind of close this out. Okay, so yeah, Marom, Ines, we are here to just kind of, if you have any kind of plan for the next steps.
I was looking at the questions in the chat, and I think one of the very first ones might be an interesting point for discussion. And the question was, I was wondering which one is more efficient, closed loop or open loop? Perhaps Marom and Til could start giving your insights on that.
I'm quite happy to pass, so go ahead, Til, and the others. Please answer that definitively.
Yeah, a very simple question, very difficult answer. I guess the first one is like, it depends, right? It depends on what you want to achieve and how you define efficacy or efficiency here. So I guess if a closed loop paradigm works and gives an extra benefit, then you might still need to consider the effort that goes into it, right? Because establishing it and individualizing it, preparing it. So in some cases, a standard protocol that's open loop might be perfectly fine, right? But I guess we'll try to further improve those protocols, and then we have to decide for each specific case whether it's worth going for a closed loop intervention or not, right? So I guess this question just cannot be answered in general, but only for specific cases.
I mean, to your point, there's a cost to the complexity, right? So we talk. I saw a recurrent theme about easier said than done, and I mean, I think obviously all the speakers here have done it to some extent. I don't know if I've ever been at a symposium where there was so much slides on what still needs to be done. You know, we're really hammering it home, and ultimately for translation, on the one hand, you think that a closed loop system could automate things. On the other hand, we know that anything that increases complexity, certainly in the clinic, is going to be a major impediment to adoption. I mean, maybe I can ask the speakers then, and I think this is also a question question I just saw from Karana and others. Where is the place loop then, right? We'll open, I mean, most of the work that's done currently is open loop stimulation, correct? And many of us have done open loop stimulation. So clearly there's a space for it, right? And so I don't know if people have thoughts about, I mean, is open loop doomed to fail, or there is obviously some spaces where open loop makes sense?
Well, so I could imagine that it also depends on the stimulation intensity. So, for example, when we talk about tES, we typically are able to stimulate at super threshold intensities, and we may be able to entrain brain activity even while completely ignoring the endogenous state by phase resetting and just using a hammer, right, repeatedly banging on the neurons, and then they will entrain after a while. While for tACS, where we're really working at the limit, at the edge of efficacy quite often, there it might be even more important to take the current state into account and thereby increase the efficacy of the stimulation, so that we don't need to go the entire way, starting our, for example, entrainment procedure at the wrong phase and need to work all the way to get it synchronized. Could also just start at the right phase and work on increasing synchronization by recruiting more and more neurons to the same oscillation immediately. So it might depend a lot on the technique, what the additional benefit of a closed loop approach is when I'm talking about oscillations again.
Yeah, I agree with that. It's a wonderful point. Oh, go ahead, Romy.
I just wanted to say, in this respect, I think also maybe what could be emphasized is, like, as we said in TMS, we have like these kind of like external markers how to like get these kind of motor thresholds, and we don't even have this with TACS or TCS. But similar in TMS, we actually also don't know whether those motor thresholds translate to other brain areas, and I think that's also a really interesting avenue to basically look at. But equally, I think this could be first done in a kind of open loop way, or like in a kind of like, I mean, like in an offline setting where you basically first investigate like different thresholds on certain like hubs in the brain, and then basically then you could close the loop once you actually also know where basically what to optimize for. I mean, that's really the challenge.
I was looking, maybe we can address some of the other questions had to do with how do we pick this brain state. So there was a question while Til was speaking whether the M1 is related to then what you optimize or define in closed loop for M1. Does it have like a particular application for cognitive function? There was some while I was talking about why functional connectivity. Do we also look at behavior? And so I can start on our side on why functional connectivity. So this is something that we have seen in this particular tasks with working memory that we know has been associated with behavior. We know that it's something that is disrupted in some conditions. Not all of them will be associated with low functional connectivity, right? So this needs to be addressed in a case-by-case scenario. And the behavior metrics, yes, we try to incorporate behavior metrics. We try to be smart about defining our paradigm so we can get as much behavioral input as we possibly can. But it's not simple because typically the behavioral that you have in the cognitive paradigms that we're looking for, you need many, many trials to be able to have an effect size that is meaningful for you to modulate. There's not many tasks out there, cognitive memory tasks that you can tweak that will give you this effect size in a number of trials that will be optimal. So Romy and I, when actually we started with this, that was exactly the idea, was behavior. And we started by looking at the NBAC task, this working memory task, and trying to model how long it would need until we see an effect of stimulation on behavior. And it was something like 11 minutes. So for 11 minutes, it's the time that you need to have some behavioral metric that we see an impact from simulation. Imagine your participants being part of this experiment. The closed loop approach is no longer an approach that makes sense because it's very similar to we'll be doing this in a lab, exploring many parameters. So I think it's about, yes, identifying parameters that you're interested, but also being pragmatic and identifying parameters that are feasible. and then take it from there. And I think, at least to us, that has been quite important. And this is why I've been looking at fMRI, I've been looking at functional connectivity and at this stage, and also focusing so much on behavior, because for us, at least at the cognitive level, it's hard to find a marker that we can optimize in real time.
So maybe to follow up on this, I think that Marom made a very good point in the beginning. So that titration of dose is like one of the key applications of closed loop systems, for example. And for example, for tMS motor threshold hunting, we've implemented that, for example, in this BEST toolbox, fully automated. Next tMS pulse intensity depends on the amplitude of the last MEP. But this is also one reason why we start always with the motor cortex, right, with MEPs, because we'll get a very nice single trial signal to noise ratio. Of course, there's variability, but we can make single trial based decisions. And for cognitive tasks, of course, this is on the other extreme of the dimensions. So you need very long assessments of task performance to get a handle on a cognitive function, how it's modulated by the often small effects of neuromodulation. And also for other regions outside of the motor cortex, if we're just interested in cortical excitability, then we need to go for tMS-evoked EEG potentials or tMS-evoked board responses for concurrent tES-fMRI. But of course, then this is always about just, if anything, a biomarker for target engagement, as Marom was saying, right? And that's not the same as a biomarker of treatment response, which might be then more closely reflected in behavioral changes. So it might be easier, but a very important first step to be able to show proof of target engagement, because that's one of the things that we are usually just assuming when we do brain stimulation studies. So we target a certain region and we hope that we have an effect on it. But if we don't, and if we don't show that we have it, then we can't even make the conclusion that stimulating that brain region has an effect on certain cognitive function or not, right? Because we don't know whether we effectively stimulated it. So both these biomarkers are important, but maybe one is easier to establish first, and then the behavioral or treatment outcome measures come into play.
I'm also thinking about what you said before about, with open loop, you can always just increase dose, right? That's the easy way to increase effect size, is just crank up the knob. And we stop at that. We don't always do that because we're worried. We're worried about tolerability, or at some point we start worrying about safety, or we even worry about side effects, which things we don't want. And so those things are not talked about very much, you know what I mean? But to the extent that those things move more to the forefront, you're improving working memory in some people, but you're decreasing it in others. Now the pressure builds, right? Okay, so we can't just increase intensity or do more sessions. Now the pressure builds to titrate. I want to be sensitive to time. I know, unfortunately, Hamid had to run to a flight, so he's not here to tell everyone goodbye, but we certainly can. I wanted to thank all the speakers for coming. I wanted to say, me and Hamid we came up with a list of sort of the smartest people we wanted to hear from, and they all said yes. So we were very happy to have you here. And I hope everyone, I hope everyone who attended found these talks as fulfilling as I did. And as I said, everyone here hopefully is on the mailing list for the tES-fMRI network to hear more about these events. Unless there's any more sort of administrative notes, I think we can close the session.
Thanks again for organizing this and for hosting this great event. I was very happy to join.
Yeah, thanks for organizing. Really nice to hear state-of-the-art work, also from basically many different angles. I quite enjoyed the diversity of the talk.
Thank you so much, and see you all soon, hopefully.
Bye-bye.
Stay well. See you soon.